happy everybody's here, and I get the pleasure of introducing Hans. Fitting that he's here for one of our 75th anniversary speakers, because he's been here for most of them. Not all of them, but more than half. Most of this more than half.
I've been on this globe for that many years.
Yeah, so Hans got his undergraduate at UC Davis and also his PhD at UC Davis. And he did his PhD work looking at heterotrophic metabolism of detrital aggregates in Lake Tahoe. That's where he did all his
Lake Tahoe.
Lake Tahoe, yeah, Tahoe. Tahoe comes later. And then he did a postdoc in New Zealand for a while where he really started working on cyanobacteria. And then he came here and continued his work on cyanobacteria really intensely through the 80s, and then switched a little bit into looking at more, well, kept with the cyanobacteria, but also started looking at estuaries. And then since then, it's pretty much been about half and half, estuarine work and this looking at cyanobacterial problems all around the world. And they are a big problem. They're a recreational problem, they're a drinking water problem, they're a global epidemic. And Hans will be talking about what we're learning about them and what we can do to make them go away.
Thanks, Nathan. I don't think we'll ever make them go away, but at least minimize them. So, so what I'm going to talk to you about today is sort of the main issues that we're facing with cyanobacteria. And let me just very quickly introduce cyanobacteria from an evolutionary standpoint. They've been on Earth for 2.5 billion years or more. They were the first photosynthetic oxygen evolving microbes on Earth. So we have a lot to thank them for, right? Like an oxygen-rich atmosphere. But we also are a major reason why they're expanding globally. So it's kind of a hand-in-hand thing with cyanobacteria. What I'm going to talk to you about today is sort of what we know about what's controlling them in terms of blooms, and you can see them over here. And sort of how paradigms have been changing in freshwater and also estuarine ecology with regard to nutrient loading. And I'll be expanding on this in my talks for the class. So, you don't need to take too many notes. Get them later. All right. So, and I also wanted to mention that, there are a lot of other folks involved in this research besides me globally. All right. So, what are they symptomatic of? They're symptomatic of us growing, increasing our population size with urban agricultural and industrial expansion. That's bringing more nutrients into water bodies. But also we're modifying hydrology, building dams and reservoirs and stuff like that. That's all playing into their playbook, and we'll talk about that a little bit. And then lastly, no talk is complete without talking about climate change, right? So I'm going to talk a bit about how climate change is impacting and accelerating some of these blooms and really presenting an additional challenge to how to manage them. Okay, so why are we concerned? They produce secondary metabolites that are toxic to everything from zooplankton on up to us. And you can see how massive these blooms are, by the way. So when they die and go to the bottom, there's a lot of oxygen consumption going on. So you get this hypoxia issue in bodies of water that are impacted, which can lead to shellfish and finfish kills, among other things. And also actually demise of some vegetation, like seagrasses, for example. Odor and taste problems are often associated with these blooms, so that's a big issue with drinking water supplies and lakes. And, the sustainability of these systems that are impacted is in serious question or problems. So there's a lot of issues and problems with them. I'm not going to talk to you about all of them. I'm going to first introduce you to the players, and they're really in three major groups. We have what we call the coccoid cyanobacteria, the little round green things that often form colonies, like microcystis. Some of you have probably seen these in blooms in lakes and even in estuaries. And then the second group are filamentous non-heterocystis. So they're filamentous but not differentiated. You can see each cell looks the same in the filaments. Some of them can fix nitrogen, and I'll bring that subject up later. And then the third group, and folks think that's the most sort of evolved group, are these heterocystis cyanobacteria. And they form these specialized cells in which nitrogen fixation is contained. It's not so easy to fix nitrogen in aerobic waters because it's an anaerobic process. So these guys are producing oxygen, in their regular photosynthetic cells, and then at the same time, they have to fix nitrogen. So that's pretty highly evolved, actually, in terms of being able to differentiate those two functions. And among these groups, there's a lot of them that produce these secondary metabolites that can be toxic. And I just want to say one thing about toxins. We call them toxins because they impact us negatively. But to the cyanobacteria, they're probably not toxins at all. They're metabolites, they're secondary metabolites that stay in the cells. And it's not until the cells are killed or the bloom crashes that they're released. So I'm sort of in the school of thinking that this has to do with metabolism of the cells itself. They're not afraid of being grazed. If they were, they would be exuding this stuff, for example, things like that. All right. The other thing I want to point out is that this is not just a freshwater problem, and it's expanding more and more into estuaries. This is a Brazilian lagoon, St. John's River, the largest river in Florida, and I think in the southeast. And Lake Pontchartrain over, if you ever, eating your crawfish over there on the shore with some beer, sometimes you can see a good bloom out there in Lake Pontchartrain. And then in coastal waters and seas, these are all from the Baltic Sea, which of course is the world's largest estuarine system, but it's also pretty high in salinity. Okay, so what's driving these blooms? And obviously nutrients are a key element, or key elements that impact growth of algae and cyanobacteria. So nitrogen and phosphorus are usually the two nutrients that are featured as being key constituents that enhance the growth of algae and higher plants. And that's because relative to the other elements in water, like sulfur, magnesium, major ions, these two things usually run out quickly. So that's why we call them limiting nutrients, okay? So that's important to remember why nitrogen and phosphorus inputs are so important. And then you can see there's lots of other things in water bodies that impact these blooms, including stratification, thermal stratification, which benefits them because they can move up and down in the water column as they please. And often they form surface blooms that'll basically shield light from organisms living underneath them, including some of the phytoplankton that they're competing with. And the other thing is by being able to move up and down the water column, they can get all the light during the day and then go down to the sediments and slurp up nutrients that are coming out of the sediments at night. And so this vertical migration is very common in these blooms. They're all adapted. Residence time is really important. That is the time for when a drop of water comes into a system, when it leaves. The longer the residence time, the more these guys like it, because actually, even though they form these dramatic blooms, their growth rates are pretty slow. So long residence time is key to their success in water bodies. And if you have a wet, we'll talk about this later, but if you have a wet spring, for example, that's putting a lot of nutrients in, and then followed by a drought, that's kind of the perfect storm scenario. The nutrients are in place, the residence time increases, and they can bloom and do their thing. I'm not going to talk about grazing and things like that, but obviously zooplankton don't like to eat toxic cyanobacteria, and also these cyanobacteria form colonies that are difficult to graze very often just from a physical perspective. All right, we've known for a long time that nutrients are involved in the eutrophication process, that is the enrichment of water bodies over time. And this is, we've been aware of this for more than 100 years. In the old days, the old paradigm was that primary production is controlled by phosphorus availability in freshwater and nitrogen in marine systems. And that held for quite a long time, actually, in part because lakes didn't have that much phosphorus in them early on. And we knew pretty early on that nitrogen was the stimulatory nutrient in marine systems. But accelerated loading of both those nutrients and a more than 50-year legacy of prior loading, particularly for phosphorus, because it can't leave the system, is leading to altered nutrient limitation dynamics. And I'm going to talk to you a bit about that because that's what forms the argument for dealing with both phosphorus and nitrogen now to deal with these blooms. So the new paradigm is that these human impacted systems reveal much more complex nutrient limitation dynamics as you go from freshwater into the coastal environment. And of course, that's a major challenge with trying to manage these nutrients to reduce blooms all along the freshwater to marine ecosystems. I'm going to start with what I call the CyanoHAB poster child. This is Lake Taihu, third largest lake in China. It's about the size of Albemarle and half of Pamlico Sound, so it's a big system. It has suffered from this nutrient over-enrichment problem for about 3 decades or even longer. Prior to this, it was actually a very desirable lake that was dominated by non-cyanobacterial taxa, like diatoms, for example, with green algae, which we like in lakes. But over the last 3 decades, it's undergone this state change. And now we have these blooms for 9 months out of the year, starting in May, sometimes even earlier, going all the way through October. And so you can imagine the problems here. First of all, it's unsightly, but this lake provides drinking water for about 11 million people in the Taihu Basin. So when there's a toxic event and it gets into a water supply, it's off limits. And there have been events like that over the last couple of decades. And of course, with the rapid growth of urban centers in China, it's not surprising that there's more nutrients coming in from point sources, wastewater, but also for non-point sources, agriculture. Activities in the Taihu Basin are also really high. If we look at the effects of nitrogen and phosphorus additions, and this is sort of our favorite stuff to do, right, Leah and Alex? We use what are called bioassays. And bioassays, in the simplest terminology, are asking the algae what's limiting by running an experiment where you collect the water from the system you're interested in. The controls are untreated, they just go back in the water, and then you have a whole series of treatments of different nutrients, combinations, and even dilutions of nutrients that will tell us what's limiting and how much it might need to be reduced. And when we see the pattern in Taihu, which is very similar to other eutrophic lakes, is that in the spring, phosphorus tends to be the stimulatory nutrient. And that's because there's a lot of nitrogen coming in from that spring runoff, okay, into the lake. But then as you get in the summer, you see that nitrogen becomes the limiting nutrient, phosphorus not so much. These are the controls, by the way, that we're comparing it to. And then later in the summer, it increases in nitrogen stimulation. Following year, phosphorus, again, is winter, spring, phosphorus is limiting. But look at nitrogen that comes on pretty strongly in the summer and into the fall. And that's actually when the blooms are most severe. But the thing I want you to really remember from this slide is that if you add both of them together, you get more than one alone. So they're both pretty close to being limiting. If you add enough of one, then the other one becomes limiting. And if you add a lot of both, well, then you end up with the worst case scenario. And I like to always point out, this is my politician slide for folks that don't like to look at data. This is bioassay results from about a 48-hour incubation, 100 mils of lake water filtered through a filter. This is with nitrogen addition. This is with phosphorus addition, a little bit less than nitrogen, but if you get them together, they give you the most. Okay, so this is always a good kind of way to present data, by the way, for people that don't like to see graphs, including myself very often. All right, North Carolina, we do these bioassays all the time, and I just wanted to show you some results that we've gotten. And these are, by the way, on estuarine and freshwater systems that have these cyanobacterial bloom problems. And you've probably seen these out in the pond in the back. We have a lot of experts here in the audience that can tell you all about bioassays too. If we look at Currituck Sound, and this is work done quite a long time ago with Liz Calandrino, who was one of my really good master students. She went to Currituck Sound to do these bioassays because the sound was getting these blooms, and you can see it quite nicely here in the remote sensing picture, of this organism called Cylindrospermopsis, which is a toxic cyanobacterial bloom former. And when she did these bioassays, you can see nitrogen stimulated, phosphorus not, but N and P together often gave you at least the same amount as N alone in this case. But if you go to the next slide, which was done by one of our IE undergraduates, Felix Evans, and she did an undergraduate honors thesis on this later, so keep that in mind. This is something you all might want to think about. She repeated this in the Chowan River over here in Coleraine. So that's upstream from Albemarle Sound and Currituck, which would be way out here. And what she found was, sure enough, nitrogen stimulated. You can't see the letters probably because they're too small, so you have to take my word for it. Phosphorus, not so much, but look at N and P together. They give you more than N alone or P alone. So from that, we have recommended that both nutrients be included in a comprehensive nutrient management strategy in the Chowan Basin. And actually, that worked pretty well early on. This is way before 2018 when we also did this work. We were able to get the river bloom-free for about almost 2 decades, and now the blooms have come back. This is a big question that we're currently addressing, that Nathan and I and others are addressing right now. Why are they coming back? It doesn't look like the nutrients have increased significantly, but something else is going on to bring the blooms back. One thing about nitrogen is you can show that there's a strong relationship between the loading of nitrogen, which you see here in terms of total Keldol nitrogen. That's all the nitrogen you can burn up and analyze in one sample, basically. And you can see that's gone up over time. And if you look at chlorophyll, and this is not even our own data, this is total nitrogen again. If you look at chlorophyll, there's also a very positive response. And this is stuff Nathan initially worked on that made us interested in looking at Albemarle Sound, because you can see there's a pretty clear relationship there over time between the amount of nitrogen that gets in and the productivity and biomass as chlorophyll. Okay, and bloom organisms are part of that chlorophyll system. If we look at lakes that have been fertilized on purpose, in other words, instead of doing it in a container, you do it in the whole lake. And if you got enough lakes to play around with, like they have in Canada, you can have a control lake and then have a lake that you add nitrogen and one that you add both. And in those experiments, and by the way, these were done also in other places in Sweden, you can see that the addition of nitrogen stimulates chlorophyll. Phosphorus, yeah, but not so much most of the time. But if you look at N and P together, again, they give you the most. So size doesn't almost matter, I think, in some of these experiments, because if you do the short-term experiment, like a bioassay, you tend to get similar results as a long-term experiment by adding the nutrients to the lake. The advantage of doing the short-term bioassay is that you're not fertilizing the lake forever and have blooms for the rest of the history of that lake. And so we published a paper back in 2016 that was titled, I think you guys have this for your reading, Takes Two to Tango. Well, you got to be old like me to really appreciate that song. It was done in the early 50s. But we wrote a paper with that title. And that's another thing to tell you guys. Catchy titles often catch readers. So, it doesn't have to be a totally off-the-wall title, but if you have a good title that catches the readers, it helps. Okay, so anyway, the bottom line is it takes two to tango with this eutrophication issue. All right, so the next question that comes along then is the managers want to know, yeah, okay, we're convinced nitrogen and phosphorus are important, but how much do we need to reduce these nutrients in terms of their input to systems in order to have some sort of control on eutrophication in these blooms? And to do that, we do what are called dilution bioassays, where we dilute the lake water with a major ion solution that just has the major ions in it, but not nitrogen and phosphorus. Okay, so you dilute it in steps, and you'll get to a point where all of a sudden the chlorophyll doesn't increase because you hit that point there where both nutrients eliminate, and you add them back individually or combined to see what the difference was essentially between dilution and addition of those nutrients. And when we look at Taihu, you can see that phosphorus needs to be reduced by at least 30%. This is during the P-limited period. And then nitrogen later on in the summer, we're talking about 50 and maybe even 70%. So those are big numbers. But keep in mind that historically, that's how much these nutrients have increased in terms of loading to these lakes, and maybe even more than what we're seeing. So I don't consider it's alarming these results. But once you start looking at long-term data with the inputs, they match pretty well with how much you need to dilute the water. Lake Erie, we've been working there for about the last 3 years on a project, pretty big project. Our part of the project is to look at nutrient limitation. And we set up shop over here in with the Ohio State Laboratory, which is right on the edge of these blooms. Really good place, the Stone Laboratory. And we've run these bioassays there. And when you run them there, I hate to sound like a broken record, but you get a very similar kind of story with nitrogen. This is in late summer, nitrogen being stimulatory, phosphorus not, but N and P together give you the most. This is later in the year, you know, when the bloom is going full steam ahead and nitrogen becomes limiting. It's the one that runs out first, okay? And if we use these dilution bioassays, you can see that I think we did a 40% reduction, which is what's been recommended by the US, Canada, by whatever state, by country. I'm at a loss for words here. By country agreement, I guess. That a phosphorus reduction of 40% has been recommended. But our data indicate that they really need to deal with nitrogen too. Can't just solve the problem with phosphorus. A major reason why nitrogen inputs may be a good way to go with a lot of these systems is that nitrogen can be lost by the system through denitrification. You've probably already heard that word. I don't know if anyone's talked to you about wetlands and stuff like that, but anyway, denitrification is the conversion of nitrate to nitrogen gas that can leave the system, but microbes can also fix it. They can fix nitrogen from the atmosphere and convert it to ammonium that can then support the growth of the algae. So what we've been interested in is understanding what the relationship is between those two processes. And if you look at the net flux, that is the net direction in which these nutrients are going, you can see that denitrification, which is over here, denitrification rates and fixation rates, the net flux tends to be negative. And that means there's more nitrogen leaving than there's coming in, okay? So that's a great hope. If denitrification can help us, at least by reducing some of the nitrogen inputs, that'll be helpful. And it's being used now in wetlands, for example, to enhance nitrogen removal and stuff like that. So, you know, phosphorus does not have a gaseous form. So when phosphorus gets into a system, it tends to stay there. It's only removed by physical flushing of the system. And even that doesn't remove most of the phosphorus because a lot of it ends up in the sediments. So the sediments tend to retain it, okay? And you know, the water flows out, but not the same amount of phosphorus that's come in is leaving the system. So you get this phosphorus buildup or legacy. And so this is kind of a conceptual diagram showing you that relationship. What we're looking at here then is the inputs of nutrients, nitrogen and phosphorus. They support the growth and eutrophication process. That process produces organic matter, which will then settle to the bottom, which creates conditions that are amenable to denitrification, for example. And so as you're loading up the system, nitrogen is leaving. Remember, less nitrogen is being fixed than the amount of nitrogen going out. Phosphorus, on the other hand, is just merely cycling along in the system and tends to accumulate in the system. So this is the rationale for why we should reduce nitrogen as well as phosphorus in these systems to give it some extra oomph in reducing the productivity and blooms. Okay, the second part of my talk is going to deal with dealing with these issues, but also in the background of changing climate. And by changing climate, I'm going to talk to you about two things. One of it is temperature warming. The other one is hydrologic changes, including more massive storms, more tropical systems, although August seems to be left with a whimper on that. But just wait. And what the impacts are of those two change processes on systems that have these blooms. We know that cyanobacteria like it hot, so to speak. If you look at the growth rates of cyanobacteria, versus other algae, you see that the maximum for cyanobacteria is usually at a higher temperature than for the other algae that they're competing with. Well, of course, we know it's getting warmer, so we're hypothesizing that there's a tendency for that part of climate change to at least benefit cyanobacteria. And if you look at lakes where there's data for a long period of time, this is from the Dutch shallow lakes that was collected by Jeff Huisman and his group. You can see that temperature has increased in the epilymnium, in the upper mixed part of the water column. Okay, and there's even maybe a hint that in Lake Erie, and you can see there's a lot of scatter there, that things have gotten warmer as well. The reason there's so much scatter is because, you know, not every year is the same. And we have temperature differences and other climatic differences going on. But this is all hinting that temperature is a important factor to consider in these blooms. And if you look at data that's been collected over a survey, going all the way from Tierra del Fuego down here at the tip of South America up to the Arctic Circle, and then plot the amount of nitrogen, versus temperature, and this is the percent biomass dominated by cyanobacteria. So, you know, the warmer it gets, the higher the percentage. You can see that as nitrogen increases and temperature increases in these systems, the cyanobacterial part of the biomass tends to increase in these lakes. Now, these are not all bad players. They're just cyanobacteria as a group in general. Okay, so in general, cyanobacteria like it hot, they like nitrogen, and they like phosphorus. If we go back to Erie, Erie is the smallest of the shallowest and smallest of the Great Lakes, and it tends to warm up more quickly. And this is just a thermal imagery. You can see that it is warmer than other systems, and that's increasing, presumably. So the combined effect of nutrient inputs from the rivers, particularly the Maumee River in the western part of the lake, with higher temperatures is probably responsible for this, in part for this resurgence of cyanobacterial blooms that we're seeing. Okay, so it's getting hotter, and there are plenty of nutrients being put into Erie. If you look at the initiation of blooms and the termination, the first day of blooms, which would be above 17 degrees, and the last day of blooms is, the first day is happening earlier in the year because of warming. The last day is happening later because it's taking longer for the lake to cool down. Okay, so the window of opportunity is increasing. And if you look at the growing days, so those would be the days where you'd be above 17 in Erie, you can see that that's increasing. The growing days, say from 2000, early 2000 to now, has increased by about, 20, 30%. So again, from a management perspective, that sort of ups the bar in terms of how much we need to reduce nutrients to get over this warming effect and the increase in climatic conditions favoring these blooms. And hydrologically, things are getting more extreme. I don't think I need to convince you all of that. We're seeing record rainfalls and flooding. Right now, the south is just pretty much covered. If you look at, there's Pakistan, half the country is flooded right now. These things are becoming more and more extreme. On the other hand, droughts are also becoming more extreme. Let's go out to the western part of this country, Australia, Africa, other places. And so with wetter and more extreme storm events, we're going to get more rainfall, which is going to bring more nutrients in from the watershed. A lot of those nutrients are man-made, like fertilizers, for example, and urban parking lots, stormwater runoff, et cetera, bringing more nutrients in. And that's impacting Lake Erie. The precipitation is more extreme. We're getting more nutrients coming into the lake from loading, and that certainly can be seen in the Maumee River, the western part of the lake. If we go back home here, there's a hint that cyanos are expanding in North Carolina's coastal, estuary and coastal waters, and that's promoted sometimes, or most of the time, by more extreme events, which would load more nutrients into these systems. And then, once it gets into a large water body, like Albemarle Sound, there's plenty of time there for these blooms to expand and grow more. And this is just a remote sensing showing the tributaries, which already have quite a bit of cyanobacterial bloom activity in them. If we go to Louisiana, the floodwaters from the Mississippi and the Atchafalaya, I think that's right, Atchafalaya River, which is over here, those floods are getting more extreme and bringing more nutrients in. So what's happening is that the blooms are expanding into a region out here, which used to be just dominated by marine phytoplankton, but now the drop in salinity and the increase in nutrients is stimulating the growth of these cyanobacteria coming all the way out into the Gulf of Mexico. So that's just another example. And I always like to show this slide because this just, I think this really brings home the issue, because Florida is a very densely populated state. But with the more extreme events that we're seeing in Florida, they're causing flooding and overflowing of the largest reservoir, or the largest lake in the southeast. It's actually a reservoir, Lake Okeechobee. And, when it gets full, the Army Corps of Engineers has to pull the plug and let some of that water out. And it happens to go in both directions, to the east here through the Atchafalaya River, and to the west out here to Sanibel Island. And with that, the other thing I should mention is that Okeechobee has this tremendous cyanobacterial bloom problem because of nutrient overloading from agriculture and urban development in the basin. So that cooks up the algae, and then when you get too much water, you have to release it. It goes downstream into the coastal waters and estuarine systems, and leads to blooms that can actually form down in the estuary or even in the coastal zone. So these folks that have these multi-million-dollar homes in these places are now seeing massive cyanobacterial blooms. In part, they're transported, but they maintain their growth because there's more nutrients also being loaded into these water bodies. All right, one interesting thing, and Rick will like this slide because it's part of his work on this system that we did. In North Carolina, we get extreme events, storm events as well. And these events, obviously, they have a lot of wind associated with them, but also they stir up the water column and mix it completely. And let me just, okay, good, this worked. So we had Tropical Storm Hannah back in 2008. It stirred up the water column, so all these parameters basically were uniform while everything was being stirred up, including temperature, salinity, DO, and chlorophyll. And what I want to point out to you is that before the storm hit, we had chlorophyll up at the surface here, a bloom of different phytoplankton, including cyanobacteria. And then the storm came along and mixed everything up. But it also caused a lot of turbidity in the water. That's the sediment resuspension going on in the system. And with the sediments, you get an additional nutrient load. So after everything is calmed down, the phytoplankton love these events because there's more nutrients in the water from this resuspension event. And you can see, based on chlorophyll, that the blooms are actually bigger than they were before the storm hit. I think there's this sort of belief that, if you get enough of these flushing events, the algae will go away and go out to sea. But it's not so simple, and they don't give up that easily. So resuspension of sediments is really important under those conditions. And also, of course, the nutrients coming in from the watershed at the same time. So it's kind of a dual whammy effect. And if we go back to Taihu, we see a similar picture. I've been going there long enough to see years where we had typhoons versus non-typhoons. And when you get typhoon years, and they always seem to occur in August there, they, of course, are also mixing up the water column, resuspending nutrients. And this is chlorophyll looked at by MODIS satellite. You can see that after the storm event, the chlorophyll is increased. When there's no storm, the system goes into nutrient limitation because there's not as much nutrient being supplied to the water column, either from runoff or from mixing. But then here are the two events later on. So it can happen in a lake, it can happen in an estuary, and I would even argue it can happen in the coastal zone. So with that in mind, I wanted to show you the ultimate challenge here that we're dealing with. And I'm using data from Lake Taihu, largely because it's good data. It's been collected for a long time. But one thing we know about things that are happening with climate change is the temperature is increasing, the wind speed is decreasing in China, and rainfall is more episodic and increasing in the system. So if you look at wind speed, it has been steadily decreasing. That favors stratification of the water column, right? Which would favor cyanobacteria because they can get over that stratification problem and not be trapped in the bottom water. If we look at rainfall, that's also been steadily increasing, particularly episodic events. And then the wind speed, as it decreases, you can see the chlorophyll is increasing in the system. And then temperature is increasing slowly. Actually, in the wintertime, it's been increasing dramatically too. That's one thing we fail to look at often is the data in the wintertime when the blooms are not there. But overall, the temperature of the lake is increasing too. So this is a huge challenge in terms of figuring out how much we need to reduce the nutrients in order to meet the bar that's changing. Okay, everybody with me there? It's the ultimate challenge. All right, so just to end up and leave enough time for questions and stuff, recommendations for nutrient management. Got to reduce both N and P in most cases, and those are not small reductions usually that are going to be needed. In many cases, we're talking about more than 30% reduction in those nutrients. In fact, in the Neuse River Basin, that's what's been recommended, 30% reduction in nitrogen input. Salinity is not necessarily a barrier to CyanoHAB expansion. I think it's just a matter of how nutrient enriched the waters are going to get. And we're already seeing that in the Baltic Sea, for example. We may need to reduce nutrients more in a warmer, stormier world, and that's changing the threshold. You know, that's a real challenge there, dealing with the new threshold when nutrients are limiting and how much we need to reduce them. Blooms like it hot, and episodic and extreme events favor these cyanobacteria. And lastly, you know, there have been suggestions by managers in the Taihu Basin and other places that, well, okay, maybe we should just reduce nitrogen in the spring and phosphorus in the summer, and that'll solve all our problems. But that's not so easy because these lakes and coastal waters have long residence time. So what you put in isn't going to go away very fast in these systems. So I always recommend you got to look at this as an annual reduction in nutrients, not just a seasonal reduction. And, you know, you got to bite the bullet on looking at this all year round, even when there aren't blooms, because the system is accumulating nutrients all the time. And so with that, and lastly, the window of opportunity is increasing. Blooms are happening earlier, they're going away later. And in some places now, like Taihu, we're almost getting them year round. So that's a real serious problem as well. So I'll stop there and just one more take-home message. No long-term study. You should not rely on one funding agency for a long-term study. You know, things change. You got to knock on a lot of doors to keep these programs going. But I think that this data, as well as other data, has shown that the value of long-term data sets in terms of understanding how things are changing, either for the better or for the worse in these systems. So that's why I put all these funding agencies in there, and you'll recognize some of them. And also, China has supported some of our research through the Chinese Academy of Sciences. And even though I can't travel to China right now, we're publishing papers and looking at data. And, you know, this is one of the nice things about the internet and communicating over long distances. So I'll stop there and be more than glad to answer any questions and for sure interact with the students later. Thank you.
We can take our questions here. I don't know if anybody's on Zoom.
Yeah, if anyone online has a question, just raise your hand virtually and you'll be unmuted.
Oh, fancy. Have I made it that clear that nobody has any questions? Oh, yes.
I don't know if this is really, I don't know if this is stupid, but can you put something in the lake to react with the phosphorus and nitrogen? And
No, that's a very good question, actually. It's not a dumb question at all. And it's being tried in some places. Flocculation of sediments along with the phosphorus and bringing them down into the sediments, holding them there. And then there's this compound called phoslock, which is a lanthanum precipitate with when lanthanum precipitates with phosphorus, it's like, locked up forever. Well, they think so anyway. And then add that to the system and you'll keep the phosphorus down in the sediments. That might work in a deep lake, or in a deep system where it's stratified so that it's always below the mixed layer. But in a shallow lake like Taihu, for example, that's been tried, and there's still enough phosphorus coming out to sustain the blooms. There are other efforts in place to get rid of the blooms, killing them, with chemicals, copper sulfate, hydrogen peroxide. There you go, hydrogen peroxide. And then there have actually been biological efforts tried, like organisms that like blue-green algae, like grass carp. But the problem with biological controls is they're not really controllable in most cases, like viruses, for example. And if you put fish in the system, you got to get them out pretty quickly too, because they're part of the nutrient cycle out there. So they're cycling nutrients around. I know of very few places where putting fish in has really had a huge impact on decreasing these blooms. The Chinese claim that in some lakes, but and there are other Chinese investigators that say it doesn't work. So no matter what's tried, still got to reduce nutrient inputs. That's always my sort of final line on that deal. Yes.
So I was really curious about the ethics behind trying to treat entire lakes, because I've seen some of the research you've mentioned has been just pouring stuff into lakes and seeing what happens. And I imagine that's a very difficult process. It's something that's very hard to get approved. So what kind of goes into that and determining if that's worth it?
Yeah, well, we're actually experimenting with that right now in North Carolina, because there's a company, I think you know this, there's a company called Blue Water, right, Nathan? Blue Green Water, that wants to treat an entire lake with hydrogen peroxide.
Right, yeah.
And, to do that, you have to get permission from all sorts of agencies, like the state agencies, but also federal agencies, for example. So right now they're going through that process of seeing whether or not they can get approval. They want to deal with Lake Mattamuskeet, and I was almost going to say good luck, but now we're sort of part of the assessment part of this.
Has your lab ever been able to get approved to treat an entire lake like that, or no?
We haven't done any of that. I've just complained about it mostly.
Yeah. It doesn't seem like a great idea, but.
Well, it might work in some places. In the Dutch lakes, for example, the problem in the Netherlands is the land has been so over-fertilized for so long that even if they stop putting any fertilizers on, they would still have 20 to 50 years of enhanced nutrient inputs. And the same's been claimed for Okeechobee, by the way. So they're, talking about dredging out the sediments, using this phoslock, for example, so that they can at least make the lakes available for recreational purposes, things like that. And they've calculated that, if they pay 50,000 euro to treat a lake and they can make 100,000 euro by charging people to swim in it, well, it's a classic Dutch thing. You're always sort of thinking about how to make money on these deals, but that's one way to look at it, especially if it doesn't look like additional nutrient removal is going to have much of an effect.
Have you done any research with how pig farms especially affect cyanobacterial? Because I know that that's a huge problem.
Well, they're certainly part of the nutrient loading issue. Animal operations, agriculture in North Carolina contributes about half the nutrients. And of that, animal operations are probably 20 or 30% of that. Still, most of the nutrients are coming from fertilizers. But we have not specifically looked at hog farms, except we have done some work with Chris Osborne at NC State, who has been able to identify different organic nitrogen sources by fluorescence spectroscopy. And he is able to differentiate some sources like chicken waste, for example, among other sources that are coming down the river. And the chicken signal is really big in North Carolina. That's all I can tell you. Stay tuned. You'll hear more about that. Yes, Jan.
So when states set TMDLs, that is the concentration of whatever nutrient per unit flow, right? So
It's the load.
So yeah, so how do we need to change how we think about those, given that the flow is changing with hurricanes?
Possibly.
Or think about.
The state has to revisit this every 5 years. So within 5 years, you're going to get a huge amount of variability in terms of loading and stuff like that. I don't know, Nathan, you may have some comments on that too, but right now we're just happy to see that they're willing to entertain a 30% reduction, although it hasn't been met. And although they're reducing some inorganic sources of nitrogen like nitrate and fertilizers, the organic nitrogen has actually gone up because of land disturbance, stormwater runoff, and other sources that have organic nitrogen in them. And this is one of our research priorities, working with Chris and others. We just got a grant funded with some folks from A&T who are looking at organic nitrogen that's coming out of landfills, all places. So that's the big question. Some sources have gone down, but the net effect has been zero because organic nitrogen has gone up. So the big question is organic nitrogen as reactive as nitrate or ammonia, which would be rapidly scooped up by the algae.
So is it still a problem that the TMDLs are not low enough?
The problem is we're not meeting the
Or we're just-
TMDL.
yeah, you're not.
Basically, yeah. I think farmers have improved their use of fertilizers quite a bit. If you look at the nitrate signal coming in to the Neuse, and also I think the Pamlico River, it's actually gone down quite a bit. So, that's progress. But this organic nitrogen issue is creeping up. And the TMDL is based on total nitrogen. So, yeah.
We have an online question.
Oh, okay.
From Kennedy. Can probably unmute herself.
Yeah. So I had a question regarding, we're seeing all these increases in blooms. I'm wondering, with your sampling and data that you've been looking at, are you seeing increasing amounts of algal toxins associated with these blooms in terms of blooms, I guess, producing toxins more frequently in the same vein?
Yeah, good question. Toxicity of blooms varies a lot. We know very little about what's controlling that. We know obviously they need nutrients to produce those the toxins. And nitrogen's been identified in the Erie Basin as being, you got to have enough nitrogen to make microcystin, for example, which is a key toxin that has a lot of nitrogen in it. I think it's about 20% nitrogen. So the input of nitrogen probably has something to do with toxicity. But more recently, there's been a paper published in Science, which is pretty controversial, that says reducing phosphorus is actually going to increase the toxicity of the algae. That was based on a laboratory modeling study, and it's now being challenged by, among other places, the Great Lakes Center and other places. So the answer to your question is toxicity varies a lot. But if you look at lakes like Taihu, and you do a regression between chlorophyll and toxin, say microcystin production, there is a positive relationship. I think you just have to collect an awful lot of data to see it. We don't know why that toxicity varies. If we knew that, that would really help a lot in terms of managing how toxic these blooms are. But there is a direct relationship between biomass and toxicity in most cases.
All right, thank you.
Yes.
You were talking a little bit about having more problems recently, like in the Albemarle Sound, but particularly the Chowan River, and maybe coincidentally, maybe not, they are seeing, particularly in the Chowan, an explosion of invasive blue catfish that are eating a lot of rangy clams in the system. And I was just wondering if those clams have the potential to do,
Yeah.
you know, to have limiting effects on
Yeah,
those.
so kind of a top-down
Yeah.
question, yeah. Maybe, this has also been hypothesized for Erie, is that removal of the, or, you know, the, what is it, Rangia, yeah, same question. One issue is that those clams are on the bottom, and the blooms are often at, you know, in the upper water column. So they're not going to have that direct of an effect on grazing. Although, you know, if the water's mixing, they could be removing biomass that way. I think that's still a question that is out there that remains to be answered.
Thank you.
And also, the blooms may be toxic, so, you know, the clams might clam up, so to speak.
Any other questions? Okay, well then you get a little bit of a break.
Okay.
Break before we meet with the students.
No, that's fine.
They can really.
That's fine. Thank you. Thanks for letting me present this.